Good morning, everyone. My name is Bill Nelson. I'm a scientist at Pacific Northwest National Laboratory, and I have been working with bacterial genomic and metagenomic data for approximately 20 years. This morning, I'm first going to talk to you in this session about just an introduction to environmental metagenomics. Some of this may sound a little familiar from some of the introduction that Marie just gave us, and some of this just may be review for you because you're familiar with the topic already. So let's get started. So what is environmental metagenomics? The goal behind it is to examine an ecosystem using untargeted DNA sequencing. And by untargeted, in this sense, what we mean is we're not focusing on a specific organism in the environment. We're trying to capture the entire community that's present in that ecosystem. That's untargeted. Okay, and as we heard earlier, the process is pretty straightforward. You obtain your samples from the environment, be it water, soil, like we're focusing on this week, air associated with animals or plants or fungi. And then you isolate the genomic material, which is usually DNA. But if you happen to be interested in studying RNA viruses, maybe you're isolating RNA from that sample. Once you have your genomic material, you're going to run it through a sequencing platform. And there's a number of them available that we'll talk a little bit about later. And then you analyze that sequence data to determine the organisms and genes that are present in your samples. And then from that information, you try to predict ecological roles of those organisms or communities from that data. And why was environmental metagenomics revolutionary? Well, this gets back to the fact that traditional microbiological techniques that have been used since Pasteur essentially require that you be able to culture your isolates. You need to be able to grow them in the lab. And you'd go out to the environment, you'd take a sample, you'd put it on a plate, and whatever grew, then you could take that back to the laboratory and you could test different carbon sources or whatever physiological studies you wanted to do on the organism. But you needed to be able to grow it first. And this greatly advanced our understanding of microbiology, but there was the issue that was termed the Great Plate Count Anomaly by Staley and Konopka in 1985. And that was that if you looked in the environment, it looked like there were more microbes out there than you could actually grow on a plate. So this is these two graphs that are on the right here. Across the bottom is month and the y-axis is depth, and these were samples that were taken from Lake Washington. And this student did two different techniques to enumerate the bacteria that were present in these samples. The top was an acridine orange direct count using a microscope, and with the darker colors being higher concentrations of bacteria. He took the same samples and did a plating assay to do the counting. And that's the lower graph here. And you can see that there's a really very large difference in the number of bacteria that are present in the environment that you can grow and that you can't grow. And so this was the Great Plate Count Anomaly. And it was very concerning for microbiologists to be saying, oh, we're studying these organisms, but are these things actually relative to the environment? And so we started getting the idea that what you could do is go to the environment, especially as we got into doing molecular biology and cloning, that you could go and isolate the DNA and then clone the DNA, and then you would have something from those organisms that you couldn't grow, that you could work with in the laboratory. And that was the first use of the term metagenomic, was for these cloning technologies, but that quickly transitioned to sequencing. Metagenomics was first used in the literature by Jo Handelsman in 1998. And in that paper, she said many lines of evidence show that fewer than 0.1% of the microorganisms in soil are readily cultured using current techniques. And most impressively, the other 99.9% of soil microflora is emerging as a world of stunning novel genetic diversity. And it's really hard to overstate how surprising and stunning it was to be able to see the more metagenomic studies that were done, the more sequencing that was done, just the more diversity was identified. And this was used at a grand scale the first time with Craig Venter's study of the Sargasso Sea. He went out on his boat, filtered a bunch of seawater, collected bacteria on the filters and prepped that, and performed shotgun sequencing using Sanger sequencing back then. And just had an explosion of diversity from this one study. So just a minor example of this is the tree on the right-hand side is proteorhodopsin genes. This is a light-driven proton pump that was discovered in Ed DeLong's lab by Oded Beja. And this was actually discovered using metagenomic cloning. And so he had done a couple of studies, also in seawater, and had discovered this grouping that was all down in yellow here. And there were some known homologs of rhodopsins in some of the sequenced isolate genomes, and those are all here in blue. This study that was not even targeting looking for proteorhodopsin genes greatly expanded the diversity of known genes, just to a surprising extent. And so this is really powerful. The amount of sequencing with the development of next generation sequencing drove this into a completely different gear. These are two graphs that are showing the content of two different databases. So this one down here is the SILBA database, which is a database of 16S ribosomal RNA genes. It started when they started collecting sequence data back in the 90s. And you can see that when next generation sequencing came along, there was just a exponential growth in the number of organisms that were detected to the point that there's 9.5 million individual sequences that are in that database at this point. And similarly, this is a plot of content in IMG, which is the representation of the Joint Genome Institute's sequencing pipeline. And it looks at both at genes that are in genome data sets and genes in metagenome data sets. And you can see that it lagged the 16S a little bit, but the metagenome projects ramped up in the early 2010s and greatly outpaced the gene discovery that was happening in isolate genome data sets to the point where in 2015, it was at 30 billion genes. And if you extrapolate that to today, now it's looking at close to 70 billion genes. So just a really powerful technique to be able to take a look at this diversity that we had no access to prior to this technique. And this extends beyond genes to organisms. Marie did mention the candidate phyla radiation, the Parku bacteria and the Roku bacteria, bacteria, all of those were completely unknown before they were sequenced. And to this day, many of the phyla that have been discovered, all these ones with red dots, are still ones that where there's no cultured representative, or at least there wasn't at the time of publication of this article. And so the power and the speed at which we've advanced our understanding of what microbial diversity is has just been incredible. Okay, so for the rest of the talk, what we're going to talk about is when should you use environmental metagenomics as a technique? What are the techniques that are available? And what are some of the ongoing challenges that you may want to keep in mind if you're undertaking environmental metagenomics? So you should remember that metagenomics is a survey technique. Except for rare cases, you are just going to be randomly sampling your community. If you happen to be in an incredibly simple community, an extreme environment where there's only a few organisms present, like acid mine drainage, then yeah, metagenomics is probably going to get you capture everything that's present in your community. But in general, the communities are going to be way more complex than you could possibly sequence to capture every single organism or gene that's present. And so this is a sampling. And so the more sequence you have, the deeper you sample, the better you're going to be representing your community. Metagenomics can tell you what's there in terms of organisms or viruses or genes, but because it's a sampling technique, metagenomics usually can't tell you what's not there, right? So just because you don't observe an organism or observe a gene, it doesn't necessarily mean it's not there. It just means you didn't sample it. And so that should be taken into account as you're evaluating and interpreting your data sets. Questions you can answer with metagenomics. You can answer questions about diversity and dynamics, which organisms are present, what's their relative abundance, what's the diversity of the community, how are the organisms distributed across space and time. You can ask questions about ecosystem function, what genes are present, which organisms encode which functions, and how are those genes distributed across environments. And getting above the gene level, you can start to integrate across genes and put together pathways and say which functions or metabolisms are available to this ecosystem. You cannot answer questions about what is actually occurring in the ecosystem because metagenomics is not looking at expression at all. It's just looking at the genes that are present. So here's an example of some global biogeography data that was collected. This is also out of the Martini lab by Michaeline Nelson. This is looking at nitrogen cycling genes and traits in soils. So she had gathered together a number of different metagenomic studies across diverse habitats and looked for the nitrogen cycling genes categorized into different nitrogen transformations and looked at their distribution in bacteria, archaea, and fungi. And also this plot over here has the samples laid out by latitude on the X and the frequency of their presence on the Y, and then the samples are all colored by their habitat type. So this was a nice study, and it shows something that you can do to look at diversity of genes and distribution of genes in the environment. Similarly, also as Marie mentioned earlier today, in discovery of new organisms, this is looking at the OD1, which I think are now the Parku bacteria, but this is Kelly Wrighton's work. I forgot to put the reference for this on here. I will do that, and I'll put that in the chat later. But here, from environmental samples, they were able to reconstruct near complete genomes. And this was something that's sort of been a dream since metagenomics was started, back in the early 2000s, was to be able to go into an environment and get whole genomes out and be able to do things like metabolic reconstructions to get an idea of what the ecological role of these organisms that were previously unavailable to scientists, and get a greater understanding of what might be occurring in these environments. The last thing I was going to say about when you should use environmental metagenomics is that it's a great support for other techniques that can tell you something about the functional expression that's going on. So if you're doing transcriptomics or especially proteomics, it can be very important to have a reference metagenomic data set to compare those data sets back against for interpretation. In terms of experimental design, you may want to do metagenomics. Make sure if you want to do metagenomics, you have a question that's addressable by metagenomics. It's always awkward when you generate a metagenomic data set and you take it to your bioinformaticist and say, hey, what does this tell me? It can tell you lots of things, and you certainly will get answers, but the problem with an approach like that is that there's a lot of data and there's a lot of different ways to analyze it, and you've just given them like an open-ended question. So if you don't have a specific question in mind when you go into this type of investigation, it's easy to get into that never-ending study. People just keep going down rabbit holes, different directions. And so you really want to put some limitations on what your downstream analysis is going to be by having a very specific question in mind. Also mentioned earlier, this is an experimental technique just like any other. It requires replication and controls in order to properly be able to evaluate the significance of your results, or just understand where the changes are. Cost is always an issue. You need to make choices about what you can sacrifice to still be able to answer your question and maintain your scientific rigor, but also keep you on budget. Sampling is cheaper than sequencing, so don't be afraid to take more samples than you think you're going to end up running through shotgun sequencing. and just figure out ways that you can screen those samples with less expensive techniques to identify the samples where it looks like the most interesting things are happening in terms of your community composition, and then target those for the more expensive sequencing and analysis steps. You should take into consideration how difficult it is to obtain your samples. There are lots of problems that arise in sample prep. So you want to make sure that if you've got samples that are going to be expensive to procure, that you practice on samples that are not expensive to procure, but might share the same features of your samples that you're targeting, just so that you can make sure that you're going to be able to get out enough biomass. You're going to be able to get out enough genomic DNA from the samples that you have. Finally, the question of how much sequence should I collect? Well, this depends heavily on a number of factors, including the complexity of your community and also the question you're asking. Generally, a appropriate response to how much sequencing should I collect is how much can you afford? That's frequently a principal driver in that, but thinking about whether you're doing a comparative study, whether you're interested in the rare biosphere, those types of things can help drive what level of sequencing data you think you need to collect. So now we're going to talk a little bit about these three techniques: Amplicon sequencing, which is technically not metagenomics because you're targeting a single gene. We'll talk about shotgun metagenomics and a little bit about short read and long read technologies. And then we'll also talk about single cell genomics. So how does Amplicon sequencing work? You start with your biomass, your microbes obtained from the environment, and you extract the genomic DNA. In Amplicon sequencing, you're targeting a single gene that you want to try and identify in as many members of your community as possible to look at diversity measures. So this requires having specific primers to whatever that gene is or genetic region that you're targeting. So you add these primers to your isolated DNA, and you amplify it using PCR to get your amplicons. Then you can take these amplicons and run them through. There's some great pipelines. Mother and Chime are very robust pipelines that you can run your Amplicon data through, and they generate either the displays or at least the data for generating all these pretty displays showing the breakdown of your organism by phyla or ordinations of your data, correlating it with environmental features. And so, yeah, Amplicon data is great at telling you who is there, and the pros of Amplicon data is it's inexpensive relative to shotgun genomic sequencing. It's very high throughput and it's very sensitive. So since there's that amplification step, you don't need a lot of biomass in order to do Amplicon sequencing. So if that's a concern in terms of your sampling protocol, is that you don't get a lot of biomass out, maybe what you want to think about is using an Amplicon approach. The cons are there are biases in the primers. If you're doing 16S sequencing, there are different regions of the 16S that you can target, and there are attempts to make universal primers against most of these sites. The universal primers are usually pretty good, but they also, there are known problems with detection of certain taxa, and I'll show an example of that later on. There are also biases in the databases. The information in the databases is information that was generated from scientific studies that have been going on, and there are biases in terms of what organisms get studied, medically relevant organisms, industrially relevant organisms. There are going to be more of those in the databases than oddball environmental isolates. So that can lead to biases when you're doing your analysis. For 16S, if you're looking at that to, sort of say how many of my organism are present in the environment, it's important to consider that there are different copy numbers of 16S or ribosomal operons in different bacteria. So if one organism in your environment's got 12 ribosomal operons and another one's only got one, then that will throw off your numbers. And so that's something you just need to consider if you're using 16S. If you decide to use some other gene for your amplicon sequencing, then you can get into issues with primer design. One of the reasons 16S is used is because of the high conservation across organisms. So if you want to look at another gene, it's not impossible, but primer design gets tricky to try and make those sensitive to all of the organisms that may be present in your community. Amplicon sequencing can be really useful for screening samples because of its inexpensive and its low cost in terms of biomass that can be input. Moving on to shotgun metagenomics. Again, you start with your organisms and extract your DNA, but this time we are going to fragment that DNA. These days, usually the fragment size you're looking for is on the order of 300 nucleotides. Since you're going to be sequencing these fragments, you need to add on sequencing adapters, and you can also add on some barcodes. And the utility of the barcodes is that the capacity of the sequencing platforms these days is very high, and maybe for whatever experiment you don't really need, the full capacity of a lane of sequencing. If you add on, if you have experiments or samples, you can add a barcode to all of the fragments that you generate from one sample, say, and put a different barcode on a different sample. And now you can mix those two libraries together. You can mix those two experiments together, generate your sequence reads, and on the far end, use those unique barcodes to segregate the resulting reads back into pools representing the individual experiments. That's known as multiplexing, and that can help you get more bang for your buck. So you can put multiple experiments in a single sequencing run. Once you've generated your sequence reads, now it's time to do your QC. We talked a little bit about this earlier. You're going to trim off all these adapter sequences that you put on when you were doing the sequencing and trim out your low quality sequences. And then you're going to assemble. And so assembly is merely the process of trying to stitch these reads back together into the original genomic sequences that you started with up here. There are many things that lead to not getting back reconstruction of a full genome. Repeat sequences usually confound assemblers. You can wind up with sequencing gaps if you've got low coverage regions. So generally you get back a large collection of contigs. There's another term that you will hear called scaffold. Scaffolds are, if you're doing paired end sequencing, sometimes you may have a sequencing gap that is still spanned by your mate pairs. So this is a scaffold is essentially a joined contig that usually has some unknown sequence in the middle of it at the junction between those contigs. But for our purposes, you can consider contigs and scaffolds to be essentially equivalent. Once you've done your assembly, you can attempt to do your binning to generate your metagenome assembled genomes, or MAGs. And this takes into account the nucleotide composition of the contigs, the read coverage. So essentially, on average, how many reads did it take to generate your contig? All pieces of DNA coming from the same organism should have the same read depth if you run through this process. And so you can take the contigs that have similar read coverage and group those together. This slide is a little misleading, showing all of the contigs segregating very nicely into their individual organismal metagenome assembled genomes. This never happens. It's never this clean. But that's what binning is segregating your contigs into organism specific sequencing to try and recapitulate your original genome. The pros of the shotgun metagenomics is that you have access to all the genomic content. And this allows you to do things like evaluate the potential function of your organisms or community. You have the possibility of generating your metagenome assembled genomes, and it avoids the amplification biases that you would see from Amplicon analysis. The cons, one is that it's much more expensive, as we've talked about, and the data analysis is much more extensive and therefore also expensive. And another one that I thought of here is that, as Marie mentioned, you can't tell from metagenomic data whether the organism you're looking at is alive or dead, or if any of the genes are being expressed or which of those genes are being expressed. So it only gives you potential, it doesn't give you actual function. Single cell genomics is a little different than the other techniques we talked about. You start with your cells again, but this time you're going to use some sort of cell sorter to get a single cell in a tube. And then that gets lysed, and because it's a very low amount of material, you need to amplify that in order to get enough genomic material to actually run through a sequencing reaction. But once you do that, you can just go through your standard shotgun sequencing after that and get out your draft genomes. The advantages to this, the primary advantage is that all of your data is going to be organism specific. You're going to get back a sequencing pool, and you know that's all from, if you got your cell separation done properly, all that is going to be from a single individual, actually. And you could have some control over what's being sequenced, so because you're throwing it through a cell sorter. The cons of single cell genomics, it's much lower throughput. There are amplification biases that have more to do with differential amplification of different areas of the genome, and that leads to low to moderate completeness values for the sequence. And what this means is how well does the sequence you get back represent the full genome that you started with. That's your completeness value. So the Rinke dark matter publication, which I got this image from the previous slide from, they had 201 genomes, and the mean completeness value for those genomes was only 40%. They did have, some that were 80–90% complete, but then also some that were a lot less complete. So the main drawback is that you get low completeness from that technique. The platforms, if you're doing one of these, at some point you'll have to decide what platform you're going to do your sequencing on. Usually the largest considerations you're going to have are, what size reads do you want to get back? Are you going to do paired end sequencing? And then just what's the overall capacity of sequence that you need back? And because of multiplexing, you could probably use any of these platforms and just request a certain, a number of lanes, or run more experiments on a machine in multiplex that has a higher capacity if you don't need that full capacity for any one experiment. So the iSeq is your lower output. You can get paired end sequencing up to 150 base pairs, and these numbers change from time to time as new chemistries come out for these machines. So don't take this as gospel. There may be new things available. You can get up to 4 million reads per run. And yeah, this is the nomenclature on the slide is a little weird, because there's how many essentially sequencing runs they can do, but the machine can do several runs in sequence. So this, you get 1.2 gig out of 4 million read individual runs. The MySeq can give you up to 300 base pair reads and outputs up to 25 megs. The next seq is getting up into another order of magnitude higher in terms of its output, and the NovaSeq again is another order of magnitude higher in its output. So just so you understand that there are the different platforms and what their different capabilities are. For the long read sequencing, the length of the reads that you're going to get out is going to vary somewhat depending on the preparation of your genomic material. But if you get good preps of it, you can get long reads out. Ion says you can get reads greater than 4 KB, and I know you can get ones that are, up to 10 KB, and you can get 50 gigabytes per run out of those. The PacBio machines, they say that you can get an average read length that's going to be greater than 10 KB if your DNA is prepared properly. And you can get up to 10 gigs for a run from the RS2 and up to 100 gigabytes from the PacBio sequel. When do you want to do short read versus long read data? Short read data tends to have lower error rates than the long read data. It's currently less expensive. Then the main advantage of short read versus long read is that you're going to get deeper sampling. So think that if you have 50 gigabytes or gigabase pairs of sequence data that you have back from short read data that's 200 base pairs in length, versus long read data, you've also got 50 gigabases, but it's 20 KB. You're going to have several of orders of magnitude deeper sampling from your short read data than your long read data. Long read data, the most advantageous thing is the longer sequence reads. You have more contiguous data rather than having on a single fragment part of one gene. Now you have multiple genes on that, and so there's some contextual data that gets preserved in long read data. Eliminates some of the complications of genome assembly. Long reads can span the repeat regions, which confound the assembler. So generally, you're going to get much better genome assemblies out of long read data. And you can also, they've got techniques for picking out methylation data as well, which is something you can't get from short read data. A combination of both long and short is certainly very powerful. You can get the assembly advantages from long read and the error correction from short read data, and easily close some genomes. I haven't seen too much metagenomic long read data out yet. I'm sure we'll be seeing more of those studies moving forward. But the primary thing that you want to consider from short read versus long read is what data do you need to answer your question? If you definitely want to make sure that you've got a deeper sampling of your community, you probably want to go with the short read data. If you really want to pick out near complete genomes for the more abundant organisms in your community, then maybe you want to go with long read data. So now let's get into some of the challenges and caveats. Pretty much every step in metagenomic sequencing, there are some biases that exist, or just troubles that can arise. So from sampling, you want to consider your method of collection, how you're taking your samples, how you're preserving your samples, what's the transport time between your sampling site and where you're going to process these samples to generate the genomic material. You want to make sure that you aren't going to run into conditions where you're going to significantly change the nature of the community that you're trying to evaluate. The size of the samples, this sort of refers to, different matrices are going to have different amounts of biomass in them, and you need biomass in order to get the genomic material out. So how much of your sample do you need in order to get out the amount of genomic material that you need is a consideration. And just the amount of processing that you're going to need to do to get the biomass that you want can affect the number of samples that you'll be able to afford to collect, or have the time to collect, or the manpower. Relating to DNA extraction from your biomass, some cells are harder to lyse than others, and this means that depending on the extraction technique you use, you either will or will not capture those cells in your analysis. Also, different extraction techniques and kits have different efficiencies. So I do have an example of this. So this is a study that was done that was comparing 4 different extraction techniques. So method 1 was liquid nitrogen, freezing the cells and then grinding them up. Method 2 was bead beating. Method 3 was phenol chloroform and bead beating. And method 4 was a purchased kit. And across the replicates, and I should explain, the analysis here is a DGGE, denaturing gradient gel electrophoresis. This is a technique where you amplify out a fragment. Usually it's a 16S fragment. And then instead of sequencing it, you run it on this denaturing gel that causes the different sequences to run to different levels depending on the sequence content. And so the bands should represent essentially different OTUs. And this is another inexpensive technique you can use to screen samples. But so they've had replicates here clustering nicely, but you can clearly see if you look at the banding patterns that there are differences in the extraction efficiency for these different organisms across the different techniques. And so the important part is to not to say, oh, I'm going to find the technique that's going to eliminate bias. As far as we know, that doesn't exist. But it does underscore the fact that you need to be consistent in the techniques that you use when you are doing these types of studies. And you may want to try, if there's especially a particular organism or clade that you're interested in, you may want to try out a couple of different extraction techniques to make sure the one that you're interested in is well represented in the data set that you're collecting. Library construction. If you happen to be interested in having insert sizes, like I said, normally these days you're fragmenting your genomic DNA down into about 300 base pairs, but there might be reasons that you're interested in having a variety of insert sizes. If you're trying to, that can be advantageous when you're doing some assembly. If you want to go from contings to scaffolds, having different insert sizes for your library can be useful. In library construction, you can get smaller fragments overrepresented because the ligation efficiency winds up being higher for those. And there may be other sequence-specific biases. PCR amplification, we talked a little bit about primer bias and uneven amplification in the single-cell genomics. I've got an example of that here. So primer choice affecting your diversity results. This was a study that was done specifically to compare two primer sets, this V3, V4 primer set, and this V4, V5 primer set in Antarctic marine samples. And this was really kind of interesting because in two of the environments they looked at, the primer sets behaved pretty equivalently. This is a Chow-1 richness measure. And so for the sea ice and the surface water, you basically got the same answer out. But in the other two environments they looked at, not only did they get different answers using the two primer sets, but you actually had different trends between how they behave differently. So in the deep water, the V3, V4 gave you a higher richness, whereas in the sediment, the V4, V5 gave you a higher richness. So again, and this is very important. If you're comparing your experiment against an experiment someone else did, certainly you need to make sure that similar techniques and similar primer sets were used before you go in and do that sort of comparison. I talked before about bias in the databases, and this slide is getting to that a little bit. So this tree here is a representation of all of the SILVA database, all of the sequences, taxa represented in the sequence database. And the branches are colored and the width is representative of how many sequences in the database belong to those different taxa. So you can see, not surprisingly, that the proteobacteria, the gamma proteobacteria, the alpha proteobacteria, the betas are all have higher representation than some of these other taxa, also the Firmicutes, Bacteroidetes, Actinobacteria. So there's not even representation of organisms or clades in these databases. And over here, this is representing or demonstrating that primers don't necessarily hit all taxa. So this was a study where they took a V4 primer pair and just computationally applied it to all of the sequences in the SILVA database. And the color is representing what was the proportion of PCR success, with the purpler colors being closer to 0. So there are some fairly diverse taxa here that just were not identified using this V4 that they were testing. Okay. There can also be biases in the sequencing platform that you use, or certainly the sequencing type that you use if you're using short read versus long read. And so again, it raises issues if you're going to try and do comparison between data sets that were generated in different ways. So consistency in your protocols. Okay. So to summarize what we've talked about in the last 40 minutes or so, environmental genomics, powerful technique to provide insight into the diversity of a community without having to culture your organisms. It's an observational sampling of your community. Various techniques are available to help you answer your question, and it can be very powerful and provide, in some cases, too much data. But you need to be sure to use appropriate techniques to make sure you maintain your scientific rigor with controls and replication, and be consistent in the techniques you apply as you do your studies. So that is it, and I can take questions.
Thanks, Bill. So we have, we're right at 10 o'clock, but we can take a couple before we sign off, and then we'll just direct to Discord / Bill, if you can chime in on Discord for anybody's additional questions. So Abigail K asks, can you provide examples of inexpensive techniques to screen samples and identify which ones should be sequenced from slide 13?
Yes, as I said, Amplicon sequencing is frequently used for that purpose. You can also use the DGGE. It's a technique that's fallen out of favor since Amplicon has gotten as inexpensive as it is. But again, it sort of depends on what your question is. It might be that you could use some enzyme assays to evaluate your samples and see what's going on in them, and see where you're seeing changes in what the community is doing, and that might be what tips you off as to which samples that you want to sequence.
Great. And then one more from Sandra Liu. If you are comparing the effect of an imposed stress like drought or salt in the soil microbial communities, do you consider that we could observe changes in the diversity or abundance of microbial communities in a short time? 2 weeks of stress, for example, or would you recommend trying a different approach?
Yeah, it's hard to know. It seems like 2 weeks would be a long enough, if that's the question, if 2 weeks would be a long enough perturbation to see changes in your microbial community. But yeah, you'd have to sort of do the experiment and find out, I guess. The only thing I'm thinking is, if you happen to have a community, if it's frequently experienced by that community, that it undergoes those drought stresses, then it might be that your community is well adapted to that condition, and you won't see much of a change with that perturbation. So it sort of depends on how adapted your community is to whatever your stress is, to see whether you'd see an effect on it in terms of community structure.
Great, thank you so much, Bill.